emphasis on value. I got my bachelor's degree in biotechnology at the Technical University of Madrid. And since October 2020, I am working in the biochemistry and molecular biology branch of the Development Americas. In this talk, I am going to present you the main results obtained by our research group from the study of the effect of NOx receptors on expression in the stochastic differentiation of mesenchyma syndicates 10T1/2 cells. This study is part of the project named NOx GL1 and GL2 proteins in adipogenesis and osteogenesis, function and mechanism of action. This project has been co-founded by the Education, Culture and Sports Department of the Regional Government of Castilla-La Mancha, Spain, supported by the European Regional Development Fund. NOx receptors are transmembrane proteins that have been described as crucial in processes such as cell proliferation, migration, or differentiation. Mammalian cells possess 4 NOx receptors, NOx1, NOx2, NOx3, and NOx4, with an extracellular domain, NECG, and an intracellular domain, NICG. These proteins are activated upon interaction with the ligands and adjacent cells. Activation results in the processing and the release into the cytoplasm of the intracellular regions that translocate into the nucleus where it binds to CSL, RBPJ kappa factor, and DNA, producing a conversion of the activity from transcriptional repressor to transcriptional activator to regulate the expression of several genes, including S1 and K1, its main target. The results obtained so far in different biological systems are contradictory. In some cases, NOx signal seems to favor osteogenesis, and in some others seems to reduce it. These facts could indicate that the role of NOx receptors in the osteogenic differentiation may change depending on its expression level and the differentiation stage of the cell. To evaluate the effect of NOx receptors overexpression in the osteogenesis of C38 cells, we generated stable transfectants overexpressing each of the 4 NOx receptors by transfecting C38 cells with expression plasmid. As shown in the representative Western plot essay in Figure 1, we obtained cell population with substantially increased levels of NOx1, NOx2, and NOx3 proteins. However, we were unable to obtain a stable significant increase of NOx4 receptor. We also analyzed the level of NOx-dependent transcriptional activation by luciferase assays. Figure 2 shows how C38 cellular populations overexpressing NOx1, NOx2, and NOx3 exhibit higher levels of NOx-dependent transcriptional activation. In these assays, cells were transiently transfected with a plasmid that had the expression of the luciferase controlled by an inducible promoter responsive to the activation of NOx receptors. The reason why this promoter responds to NOx activation is that the plasmid contains 4 CBF binding sites, acting as an enhancer of the luciferase transcription. In absence of NOx intracellular domain, CSL/RBG kappa is joined to a co-repressor, but when NICG translocates to the nucleus, this protein complex gets dissociated and CSL/RBPJ kappa bind to NICG, promoting the expression of the luciferase. Cells were also transfected with a plasmid that enables them to express renin constitutively to correct luciferase signal variations due to the number of cells. Likewise, in Figure 3, it is shown the mRNA levels of 2 of the target genes of NOx activation, S1 and K1. As expected, we observe an increase in their expression in response to the overexpression of NOx1 and NOx3. However, opposite to what we expected, NOx2 and NOx4 transfectants showed a reduced expression of both target genes. Anyway, These are preliminary results that must be biologically replicated. The next step was to evaluate the effect of the overexpression of these receptors on the osteogenic differentiation of CD8 cells. For this purpose, osteogenic differentiation assays were carried out. Figure 4 shows the result of alkaline phosphatase staining of osteogenic-induced cultures at different days of treatment. A higher intensity of staining was observed in NOT1, NOT2, and NOT3 transfectants compared to the control population. The intensity of staining is an indicator of the degree of osteogenic differentiation. On the other hand, in Figure 5, the mRNA levels of 4 characteristic markers of the osteogenic differentiation process in this cell's population are shown for different moments of the assay: days 1, 7, 14, and 21. Alkaline phosphatase, referred as ALP, is an early marker of the differentiation. A significant increase in its expression was observed in every overexpressing population. Osteopontin, referred as OPM, is a late osteogenic marker, but it is also activated in earlier stages to inhibit cell proliferation. A significant increase of its expression could also be observed at day 14 in cells overexpressing NOT1, NOT2, and NOT3 receptors. RANX2 transcription factor expression has been described as a crucial element in the cell fate determination. Its expression showed also a significant increase at day 7 in C38 NOT1 and C38 NOT3 transfectants. However, no significant increase in collagen, referred as COL1A1, was observed. In conclusion, our results indicate that C38 10T1/2 cells can stably overexpress NOT1, NOT2, and NOT3 proteins. Stable overexpression of NOT genes in C38 cells increases NOT receptor-dependent transactivation levels. Overexpression of NOT receptors modulate HES1 and K1 expression levels. Overexpression of NOT genes in C38 cells increases alkaline phosphatase staining, and the stable overexpression of NOT1, NOT2, and NOT3 receptor in C38 10T1/2 cells enhances osteoblast. Thank you to everyone for attending this talk, and thank you to every member of the research group I have the pleasure to work with.